Thank you very much, Hannah. And you're being recorded. So thank you very much to Hannah for that nice introduction. Thanks to Rachel for inviting me, and thanks to all of you for showing up. As I said, please, you're welcome to interrupt and ask questions, but I also understand that they'll be recorded and asked at the end. So this title of this call, I called it Methane in the 21st Century, a case study of plastic in the marine environment. Building on some work that we did a couple of years ago now that attracted quite a bit of attention. Here. So why the interest in methane, and sort of why the interest in methane and why the interest in its sources. So if we think about methane globally, on the left-hand side, we have atmospheric concentrations of methane as they're being measured at the Mauna Loa Observatory on the Big Island of Hawaii. Concentrations continue to increase. They're now almost approaching 2 ppm in atmospheric methane, so much higher than what we would like it to be. There was a bit of a pause around about 2004 to 2006 where we thought maybe it's not such a big issue, but since then concentrations have continued to increase. And there's a really nice working group called the Global Carbon Project that's highlighted on the right-hand side, which for all of the greenhouse gases, carbon dioxide, methane, and nitrous oxide, they produce these nice schematics which summarize the sources of methane, which are divided into orange, which are the anthropogenic fluxes, and green, which are natural fluxes. So some of the anthropogenic fluxes being fossil fuel use and agriculture, wastewater, natural fluxes being from wetlands, oceans, and other natural habitats. So we've got a fairly good grip on the global methane budget, the sources and the sinks. And historically, if we go back 20 years ago now, we would have thought that the methane, our knowledge of methane, is sufficient so that we can complete the budget. So on the left-hand side, a fairly busy diagram showing that in reality, methane is formed. Majority of methane was being formed then under anaerobic processes, where you have the decomposition of organic material, ultimately leading to the production of methane, and then methane would being oxidized, and that's shown by the dashed orange arrows. So 20 years ago, people weren't looking for unknown sources of methane. It was generally considered that if you that we had a sufficient understanding of the major pathways, and if you wanted to start looking at cryptic cycling of for any particular element, actually the nitrogen cycle shown on the right-hand side with a much greater, I always think the nitrogen cycle is much more susceptible to cryptic pathways, unknown pathways, as you've got a lot more arrows occurring with the nitrogen cycle versus methane, which really just had one major source and one major sink. But in the open ocean environment, there was always a phenomenon that was not well understood. And this phenomenon was that methane concentrations were quite often highest very close to the ocean surface or very close to the surface of a lake where it was an aerobic environment. And that was confusing because methane production predominantly is an anaerobic process. So if we were to consider a water column, you'd expect that methane to be highest, closest to the sediment, because that's the closest proximity to an anaerobic habitat. And you wouldn't really expect to find any significant methane concentrations in the aerobic environment. But whenever scientists would go out into either a lake environment or to an oceanic environment, they'd quite often see this subsurface peak in methane, which wasn't really well understood. And that subsurface peak in methane is shown by this dashed line, which has a peak just above that yellow box, and the yellow box is referring to oxic methane production. And for many decades, that was referred to the methane paradox. And the methane paradox was that, if it was summarized, methane is thought to be an anaerobic process, yet there's this subsurface peak in methane in an aerobic environment. So presumably, people thought that there were microenvironments, such as the guts of zooplankton, or particle material, or fecal pellets, which could host these anaerobic microniches. That's generally was the assumption that was sort of going on there. However, a little under 20 years ago, two scientists, Dave Carl in Hawaii and Frank Kepler in Germany, really opened up the black box on that, and they both reported aerobic methane production. Dave Carl reported it for the ocean, Frank Kepler reported it for terrestrial land plants, and really just said that methane isn't just limited to anaerobic bacteria or anaerobic archaea, but rather there's a wider variety of organisms on the planet that can produce methane. And those two landmark papers, let's say in 2007 or 2006, really came under a lot of scrutiny from the scientific environment, and a lot of people didn't initially believe them. But fast forward to the current day, and now it's known that there are multiple pathways for aerobic methane production, and this has been validated by the scientific community. So if you Google it, you'll see everything from reactive oxygen species, which can produce methane, methylated compounds, so CH3 compounds can produce methane, and there might also be some cryptic photosynthetic related pathways, in addition to the reactive oxygen species. And really that sort of that box of everything from a zebra to a tree to a phytoplankton and a bacteria highlights that methane production is, to some people, is considered ubiquitous to life on the planet. In the oceans, we tend to still think of it more as limited to methylated compounds. So in this instance, the example that I've given is if you have a methylated methyl phosphonate. So it's a source of phosphorus connected to a methyl group, CH3. The bacteria snip, that's shown by the scissors, they'll snip that bond. They will incorporate the phosphorus and dump the carbon as methane. This was something that Dave Carl investigated and continues to investigate in Hawaii for many years. And the back garden to the University of Hawaii is the North Pacific Subtropical Ocean, shown in the top left. The catamaran is the research vessel, the UNOLS research vessel that's operated by the University of Hawaii. Both Rachel and I have sailed on that many times. And what my role was to investigate the role of the impact of light on this aerobic methane production in the surface ocean. So the schematic in the top shows sort of this sunlight so in the search, just to quickly orientate ourselves, it's not like the northern hemisphere of Europe. There we have sunlight reaching down to almost to 200 meters. And in warm, so 25 to 27 degree water, and in that sort of low nutrient environment. And to investigate any biological process in this environment, scientists love to collect seawater from any particular depth. We will stick it into a bottle that's shown on the right-hand side, and we will hang those array of bottles off a buoy, and it will just be suspended in the ocean for as long as it takes for us to investigate any particular rate. So when I did that, looking for the potential for biological methane production, I was starting to see these sort of this is shown in the plots in the bottom hand side. This is from stretching my memory. I'd see that these control there was I could measure methane in controls, presumably that was something like distilled water, and compare that to live, which was biological, which was just natural seawater. And if I look at the percent difference on the bottom right hand side, I'd say, well, okay, maybe there's a difference between this percentage difference. When I looked at that, I was like, well, that pretty much to me resembles a light curve, the attenuation of light with depth. So I thought there would be an impact of light on this methane concentration. But the issue came when our controls, when we repeated this, our methane in our controlled samples was just as high as our treatment samples. So this was really the first step where we started to say, well, okay, methane is increasing in our control samples as well as our treatments. So therefore, it's this contamination of methane that we started to look into a little bit more detail. And our very first experiment was to collect different types of plastic, which are listed on the left-hand side, and incubate it on the roof of our building, and just to see how much methane is coming off from these particular samples. So it's an incredibly straightforward experiment, and that we used our temperature controlled system was a plastic paddling pool. That was my daughter's at the time. We just used it to control the temperature. And what surprised us was the amount of methane that is produced in some of the plastic that we investigated. In particular, low density polyethylene, or LDPE, had 10 times the amount of methane produced in some other types of plastics. And when Rachel asked me to give this talk, I went back to my notes, and this was how we summarized it. This is exactly what myself and colleagues wrote. The production of methane by certain plastics is intriguing. We didn't know whether plastic is trapped in plastic and is being leached, or whether it's continually being produced. And also, there are other hydrocarbons also being produced. And we mentioned that this was us just summarizing this quick experiment as, wow, there actually is measurable amounts of methane being produced by plastic. So what do scientists do? Well, this is a rainy day project. We probably waited a couple of months, then we repeated it, and we repeated it with these things called pellets, plastic pellets. So I don't have a massive understanding of the plastic industry, but my understanding is that these plastic pellets can then get molded to whatever item of plastic needs to be produced. And so we got hold of some of these, and in this instance, what we did then is just repeat this and look at the amount of methane that's coming off. But then we also extended that to look at other hydrocarbon gases, so ethylene, ethane, and propane. And what we found when we incubated this plastic, pretty much in the same conditions on the roof of the building, that even after half a year, the gases are still giving off. The gases are still being given off, and if anything, there's almost like an exponential increase for some of these gases as well. So it's interesting to note that methane isn't the biggest gas given off; it's ethylene. Perhaps, in hindsight, that's not unsurprising, given that it's low-density polyethylene. It's perhaps not unsurprising that ethylene is the biggest gas given off. Ethylene is also really important as a hormone for ripening fruit. That's one of its major purposes, industrial purposes. And we also have propane and other gases given off. The other aspect as well is we can return back out to the ocean, and this is not plastic part. This isn't plastic pellets anymore. This is what we call aged plastic. But you can go out into any environment, whether it's the coastline, you can find some plastic on the beach along a coastline, or collect it from the ocean. you can identify it, and that's shown in the bottom plot where we compare virgin low-density polyethylene, that's the pellets, with aged, that's it, that's plastic we find from the environment. And you pretty much find that any plastic collect or any low-density polyethylene collected from the environment, which has an unknown age, but if you collect plastic from the middle of the Pacific Ocean, it's probably been there for several years, is still giving off these hydrocarbon gases as well. So it's not something that's limited to fresh plastic. Production of hydrocarbon gases is pretty much ubiquitous to all plastic, and it's in particular it's this low-density polyethylene, most likely due to its the hydrocarbon bonds that are used to make the plastic. And you know, from a plastic and methane perspective, that's pretty much where this work was left. You know, this is a snapshot of this was sort of our initial conclusions of the work, and it's pretty much how we concluded that set of experiments. You know, methane has been, but now we are completely certain that methane is being produced by certain plastics. We don't really know what the implications are. If we scale up our do back of the envelope calculations, it's probably not that important from a global methane budget, but it could be important for global budgets of ethylene because of the amount of ethylene that comes off. But you know, are the hydrocarbon gases, you know, are they being used by microbes as an energy source or as a carbon source? And how does this relate into our understanding of microbial colonization of plastic? But we sort of concluded the work that if we think about methane being produced in the environment, you've sort of got different categories of it that make it quite difficult to understand. There's always some low-level contaminant methane. Methane in any incubation experiment is never zero. You've always got some low trace amount being produced. And then we have what we refer to as traditional methanogenesis. That's the anaerobic production of methane. On top of that, you have photochemicals, so abiotic production of methane. And then finally might be this photobiological or aerobic biological production, which is also increased by light. So methane, understanding methane is no longer straightforward as sort of measuring it in aerobic and anaerobic environments, but rather as these multi-sources that make it quite tricky to understand what's going on. When I look through the microplastic literature is now booming, both in the scientific literature and also from many articles on the internet about it. The ones that are probably most related to the production of these hydrocarbon gases by plastic is the identification of microorganisms that colonize the plastic. If you put anything in the ocean, it's going to get biofouled, it will get colonized. And so scientists are now understanding the microbes that colonize the microplastics. And that could really be looked at with regards to these greenhouse gases. And then the other aspect as well is the interesting concept that it's not just gaseous carbon that's coming off, but also dissolved organic carbon that now becomes available to the marine microbiota. So if we think about dissolved organic carbon, which is this, you know, is it a labile or is it a recalcitrant pool of carbon? Then how is this disrupting sort of marine heterotrophy metabolism? So I think that's when I look through the literature about how our knowledge of hydrocarbon gases and greenhouse gases being reduced, it's really with respect to these this sort of related work, which as I see is sort of how our insights have been, how our insight has grown. This is just, but it was reassuring because one outcome of this work is it's actually very hard to measure low-level concentrations of methane. This is something we try to do. Many countries around the world, we're trying to ascertain how good are our measurements of methane, and we couldn't understand why. For example, if we look on the left-hand plot, each different color symbol is a different country trying to measure methane. And you see, whilst there's agreement between 5 to 6 countries, after that it starts to increase. And now the work from plastic really explains why, and we realize we have much better handle that actually low-level concentrations of methane are really susceptible to contamination by and that's due to sort of storage procedures that we use for these samples as well. So a nice indirect outcome from this work. With any piece of work, there's always, knowledge that gets published and sort of knowledge that doesn't quite make it into the publication. And this is something just to highlight that, if I was to do it again, I've highlighted the hydrocarbon gases, methane, ethylene, ethane, propane that were measured, but there are other gases as well that we sort of had a bit of an understanding about, but not enough of an understanding to make it into the literature. And carbon monoxide was one which I always intended to return to it and do more work on it, but I've just, intended to return to it and never have. But there's this curious phenomenon that carbon monoxide is just pouring off of plastics, and carbon monoxide is actually a fuel source for, and that's shown here by Marianne Moran and Bill Miller, both scientists operating in Georgia, actually have commented about how there's a wide variety of bacteria that contain the genes for metabolizing carbon monoxide, yet there's not that much of it around. So this, I think, it would be another gas that would be particularly interesting to look at, which has known links to sort of photochemistry and photoreactions. So maybe at some point in the future, with an appropriate funding stream or an interested individual, this is something that I'd go and look at and again in future, because I think there's definitely more to be discovered about this in relation to marine bacteria. So stepping back a little bit and thinking about methane, so with climate change, this is, 20 years ago, nobody was thinking about aerobic methane. And 10 years ago, people weren't even really thinking about too much about methane. It was really much the neglected younger brother or sister when it came to climate change. Everybody was about CO2. That's now changed. And even recently in, I don't know, last week's Science issue, they're saying, look, we're going to completely overrun all of our targets on CO2, but there's still hope for methane. And methane is important because of its global warming potential and its shorter lifetime span than CO2. So if we look at some of our, if we look at some of the industrial or anthropogenic sources of methane, which ones of those can we really decrease quickly? Whether that's capture at the fossil fuel stage, whether it's agriculture, whether it's wastewater, how can we, there's this hope, because we don't always understand enough or that much about methane, that maybe we can mitigate its release into the atmosphere much quicker. And more technology is coming online. So again, this was from Science last slide was Science last week. This is right now, with satellites going up into the, wherever satellites go, up into the satellite world, they can now do remote sensing of emissions of methane. And that will really put the spotlight onto gas pipelines and refineries, because for the first time ever, you don't need to rely on plane or aeroplane-based observations or drones. It's satellites circling over that will be able to spot where these leaks are coming from. So that's also a cause for optimism. With myself, I'd actually like to return to my ocean, return to the some of the ocean stuff that we was doing. And next year, actually in Easter 2024, I'll be going to look at some of the methane seeps off the Turkish coast. Turkish has a lot of fault lines, which are natural emissions of methane. And when you look at this, as those fault lines extend off the coast, so we have bubbles of methane being emitted. And what I'd like to understand are, what are the methanotrophs, the bacteria that oxidize this methane? Is there, how do they compare with methanotrophs in other environments? If I compare methane emissions from from agriculture or wastewater or landfill or the ocean? Is there like one super methanotroph, or are there a suite of methanotrophs? And how can we increase their activity? Are there some nutrients or that are critical to their metabolism, such as copper, which is the metal enzymatic cofactor? Are they copper limited, or are there other microbes that they like to hang out with? So, microbes rarely act solely or independently by themselves. I'd like to know, what are their favorite cousins, favorite family members, favorite friends that are hanging out, and that we might be able to look holistically at increasing this microbial biofilter for methane. So that's where my work is heading next in the ocean world. And so it just remains for me to thank my collaborators for the methane and plastic work, Dave, Carl, and Sarah, who are still still working at University of Hawaii, Sarah Jane Royer, I think she's in San Diego now, and myself, who's now at Newcastle, and our sponsors who funded this work or related work that prompted us to do sort of more on this. So I will stop sharing my screen now and say thank you very much.